Hello, everyone, and welcome to today's live broadcast, Precision Medicine, The Way Forward, presented by Prasun Mishra, founder and CEO of Agility Pharmaceuticals, CEO of American Association for Precision Medicine. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom and/or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now please join me in welcoming Dr. Mishra. I will now turn the presentation over to him.
Thank you for that kind introduction, David. And hello, everyone. Today, what I will try to do is I'll try to give you overall bird's eye view of the precision medicine field, what we have learned, what we are trying to, what is the overall goal, and what are the problems that we are trying to solve, and and what is the vision, and how much we have accomplished. So these are my disclosures. I do not anticipate discussing off-label use of any commercial products in this educational activity. And this presentation reflects my personal views and does not necessarily reflect policies of the companies that I have founded, funded, advised, or own stocks in. So this is a brief outline of the talk, and I'll walk you through it. So first, I'd like to introduce a few key concepts. So I would like to begin with past President Obama's quote. And that's why we are here today, because something called precision medicine gives us one of the greatest opportunities of new medical breakthroughs that we have ever seen. Historically, precision medicine, evolution began with starting off pharmacogenetics. So we all studied the inherited differences in drug metabolism, and genes involved in drug metabolism and response. And with the rise of omics, pharmacogenomics came into picture, where we studied all the many different genes that have been used to determine drug behavior in order to improve the efficacy of drug. What I have, there you go. So what we have, the field, of individualized medicine or tailored medicine began, and the name personalized medicine came into picture. And what I call this as a clinical application of pharmacogenetics and pharmacogenomics. Although the name was very personalized, but we never intended to develop, drugs or medical devices for each patient, but rather, the ability to classify individuals into subpopulations that differ into their susceptibility to a particular disease or their response to a specific treatment. So, it was called precision medicine, which is a widely accepted name right now. Precision medicine is an emerging approach for disease prevention and treatment that takes into account people's individual variations in genes, microenvironment, and lifestyles. Again, precision medicine takes into account folks' family history, medical, genetics, lifestyle, and environmental factors. So why precision medicine? Today, most medical care is based on expected response to an average patient, and we know that there is no average patient. There's no such thing as an average patient. So we want to move away from that paradigm to a medical care based on individuals' genomic and environmental and lifestyle differences, enabling more precise methods to prevent and treat diseases, actually. So that preventive and therapeutic interventions can be concentrated on who will benefit and sparing the side effects to those who will not. So there are, many classic examples of precision medicine. It's not the concept is not entirely new. Starting from the testing for BRAF V617 mutations before treating with vemurafenib to testing for disease-causing mutations in BRCA1 and BRCA2 genes, which are implicated in familial breast and ovarian cancer risks. And this is and there are many successful examples, but historically, this was sometime around 2011. National Research Council for National Academies, was given a task to kind of, examine the field of precision medicine and suggest their recommendations to, where we should be going. And they recommended to develop a knowledge network. So this will be data from basic sciences and discovery, as well as clinical discovery, funneling into information commons. So this will be, molecular characterization data and EHR, electronic health records data. They will be all flowing into the knowledge network where with the various layers of the classifiers with genomics, microRNomics, epigenomics, and then using the integrated genomics omics approach, we will create a taxonomical classification that will be used again to fuel the basic research and clinical discovery. So this is the fabulous vision, but there are certain scientific challenges that we have faced, and we are solving them. In cancer field, one of the first problems is tumor heterogeneity. As you can see here, a patient can have many different lesions. So those lesions, can arise from different cells, colonizing a tumor. Also, if you look at one tumor itself, the tumor itself is heterogeneous. It's made up of different sections that may have different genomic profiles. So this is the first problem. The second problem is tumor evolution. If you see, when tumors begin, they have different, so the driving genes are different. And the mutations that drive relapse, they are presented at a low frequency. And when the tumor relapses, those mutations become predominant. So how do we treat, for that, so that it leads to the anticipation-based chemotherapy? Also, sheer number of mutations in cancers are very high. Just you can see lung cancer as an example. Non-small cell lung cancer has 147 mutations. Lung cancer, small cell, has 163 mutations. So there are a lot of mutations to target. And also mutation rates across the cancers are not uniform. You can see melanoma being the highest, highly mutated cancers, and the neuroblastoma being the lowest cancers. And in melanoma, you can see the C2T mutation is predominant. Maybe this is UV signature. And in lung cancer, you can see C2A is predominant, which may represent a smoking signature. So, as we have learned more, our view of cancer has changed. Just to take an example of lung cancer, traditionally we thought that there are three different kinds of lung cancer, right? Squamous, large cell, and adenocarcinoma. Then in late 1980s, we learned the role of KRAS, and in 2004, EGFR. And in 2009, we learned that there are other genes that were involved: BRAF, MET, AKT, PI3K. But now in 2017, there are many other genes that drive this, and that could be druggable but there are a lot more needs to be learned yet about this particular cancer. So as we learn more, we realize that cancers are more complex than we think. So also the potential of non-coding regions. So, this is all we talked about right now is the coding. So as we learn from human genome, we are not the coding region. If you see presented by protein coding genes, is a small fraction of a human genome. Rest all is repetitive DNA. Line and sign elements, also pseudogenes, and if you see, majority of the human genome encodes unique non-coding DNA thought to be a junk DNA, but this non-coding DNA encodes microRNAs and all sorts of non-coding RNAs, which may be very important, in our regulation, in regulation of the protein coding gene. Also, when we grew up from when we evolved from a simple to very complex organism as human beings, you see the 3' UTR length of a gene, a protein coding gene, did not increase in length. However, UTR, 3' UTR kept on growing, suggesting that proteins, humans are humans because they are regulated very well. The genes are regulated very well. Genes have longer 3' UTR that allows more innovation and more regulation to happen using the non-coding RNA. So, I studied this phenomenon in detail, and we put forward one schematics that's well accepted in the field now. So you have a microRNA variant that's present in the 3' UTR of a gene, and that can knock off a microRNA binding site or can create a microRNA binding sites. And this, this may result in overexpression of a drug target or oncogene. In classic wisdom, oncogenes are the drug targets, and overexpressed drug target can result in drug resistance and result in cellular transformation since it's an oncogene. And since the microRNA regulates oncogene, it will be a tumor suppressor microRNA. And this field in the last 10 years or so has exploded, and there are approximately 2,700 articles in PubMed suggesting that there's some sort of variants in different genes that drive different diseases. They affect they somehow affect those genes that drive those diseases, and by creating or knocking off a microRNA site, resulting in disease predisposition or preventing the disease to occur. So the microRNA polymorphisms are a key variant that are encoded in the 3' UTR non-coding gene, which is usually avoided in most of the sequencing operations. And the goal is that some of these variants will be populated in ClinVar or ClinGen, which is a consortium of to create actionable genomic information to clinicians at the point of care. So, and one more challenge that we see is if we have an oncogene that drives malignancy, if we try to block it, what happens is there's another oncogene, gets activated after some time. So we call it oncogene bypass. And then the combination therapy is the way to go, so to find which oncogene has been activated, and then we combine it with a different drug. And also one of the challenges that we have been facing is poor accuracy and reproducibility of lab-developed tests. If you see the example, in one of the first examples, the accuracy of this test, which was performed in 91 labs, is approximately 75%. So there is still room for improvement. So the way forward. There is a good news that we have cost of sequencing of genome has gone down due to advances in sequencing technologies. Now you can sequence your gene for approximately $1,000, and the time of sequencing has been reduced drastically from human genome being 2 years to now less than a day. And to computing power, computing power is more than 16 times more than what we had when in 10 years back, actually. And so again, the cost per genome has defied Moore's law, and then you can see that it has gone down dramatically, and to our benefit. And the technology itself, the sequencing technology itself has evolved, and now what we have done is we have sequenced a lot of reference genomes to have a very good consensus reference sequence. So if we sequence a new genome, we can align and we can get a good sense of what are the a good sense of the alignments in which maybe the region which is which variant is important versus and which is just a noise actually. So and these methods are right now in the clinic. So this is a slide from MD Anderson Cancer Center. You can see where patients are profiled based on the pathology reports or sequencing information, and based on that, they are prognostic markers predictive drug sensitivity and resistance, and markers to predict adverse events are defined, and based on those markers, patients are stratified in different groups and assigned a different treatment regime, which is very encouraging. And also another example of how to what are the cases that they are seeing in the clinic. So you have 3 different patients that are are presenting themselves in the clinic. One has a melanoma with BRAF V600 mutation. Second patient has a colorectal cancer with BRAF V600E mutation, it's the same mutation. And third patient has a colorectal cancer, but a different BRAF mutation. But the mutation is in the same gene, BRAF, which is 594G mutation, B594G. So based on type of cancer or type of mutation, right now we can prescribe different medications. For melanoma, it would be vemurafenib plus cobimetinib. For colorectal cancer, V600E mutation, it would be vemurafenib and cetuximab. And for the D594G mutation, could be trametinib or cobimetinib. So this gives you one example of gene invariant analysis in the context of each patient's disease. So moving on to the integrated data analysis. So National Research Council, National Academies recommended to create an individual-centric information commons, and they gave an example that Google Maps, there are different layers, GSI layers have been organized, for geographical positioning. Can same thing be done for the genome? Where exomes, genomes, epigenomes, microbiomes, and all different layers can be put together to generate a better picture of the patient disease. And this is something that is a current focus of my research, where we are trying to integrate multi-omics clinical and real-world data. And the goal is to create a knowledge network where we would be, which we can utilize to, develop meaningful drug diagnostics combinations, so that the right drugs can be given to right patients and at the right time. When our goal also is to accelerate drug discovery process, so time is a big factor here. So this is just to give you one example where we have put together 2 different layers: a microRNA layer, expression microRNomics, and the genomics. So this is in melanoma patients. So in this particular melanoma patient, it was driven by RAS, PI3 kinase, and MCL pathway. This pathway was over represented in these in those tumors. And we have a microRNA. We found that miR32, the microRNA, was underrepresented in these tumors. And when we overexpressed the microRNA, we found that the tumor volume in mice went dramatically down. So that was very encouraging. And this is, and what you see represented by V is the vemurafenib alone, which is a drug to treat melanomas. That also reduced tumor volume, but what was very impressive when you see miR plus V, so that's the combination where we combine microRNA expression with the vemurafenib treatment, and we saw very dramatic reduction in tumor volume. And so this also opens prospects for microRNA expression and targeted therapy combination, which is very encouraging. There are data integration, and so there are many challenges, as you can see from the slide right here. Data is, in patient data in the clinic is fragmented, actually. So we have to find a way how we can integrate that data to create integrated data sets for personal health. This is one of the challenges that we should be, as a community, be addressing. And another challenge is the data, as you can see, is siloed in different institutional firewalls. And so in our field, the data is considered as sacred. So then, how to form those consortiums or network sharing models, which will allow data to be shared. One of the examples is GA4GH, Global Alliance for Genomics and Health, that allows to share data. And also, one more problem is how to foster more collaborative research work between academia, biotech, pharma companies, with some sort of shared IP, some sort of consortium governing shared IP. So these are some of the problems that we should be working as a community to solve, so that we can make a great advance in very in a very faster way as a community. So the big data ecosystem is too big. You can see, so the slide presents, you can see there are a lot of infrastructure companies, there are analytics companies, there are also, there are applications company. This is where the life science pharma is, pulled in. There are also security companies. There are a lot of companies that provide security, which is very important. There are also, open source algorithms that you can utilize to conduct your research, and there are data sources and APIs that are available, and you can use that for your research too. But moving forward, affordability and cost of precision medicine will also drive individual decisions. So we have to also find a way to find how we can reduce the cost of the integrated omics analysis, and also how to make the payers, convince the payers to pay for these analyses, to let the patients afford promises of precision medicine. So the next is expected benefits of precision medicine approaches at different stages of clinical care pathway. And, precision medicine can be utilized in early clinical development to identify patients who may respond most appropriately to a particular therapeutic, and also to, also in recruitment and enrollment, enriching study population using predictive biomarkers. And also post-approval, the precision medicine can be used to optimize treatment decisions to ensure that the right drug can be used to treat the right patient at the right time. So, and so all three stages, prevention, diagnostics, and treatment, precision medicine approach can be utilized to improve health-related outcomes, to improve the health overall, health-related outcomes of patients. And precision medicine-based drugs have a high likelihood of getting approved by FDA due to their ability to distinguish responders from non-responders or toxic responders. And this is just to show you that in 2007 alone, over 30% of new drug approvals were personalized medicine, some sort of biomarker components associated in it. And this is a link to FDA's, FDA's unique drugs available with pharmacogenetics and label. There are approximately 240 drugs that are in the list, and this is a link. If you're interested, you can take a look at it. And last but not least, healthcare stakeholders and their role in success of precision medicine. So there are many stakeholders in the precision medicine field, and they have all their responsibilities. So governments, policymakers, research institutions, biomedical science community, pharmaceutical industry, consumer groups, regulatory bodies. So we all have to come together and work together to deliver the promise of precision medicine. And to do that, we have created an organization. It's a nonprofit organization, American Association of Precision Medicine. The goal is to reduce adverse drug events by accelerating the field of precision medicine through research, education, communication, and collaboration to foster new medical breakthroughs. And AAPM also facilitates a dialogue between the four P's of precision medicine: patients, providers, public health planners, i.e., government, and payers, to work together to achieve the goal of delivering improved outcomes at a reduced cost. And so we foster dialogue. We organize meetings and networking events to get folks together in a room and facilitate innovation, communication and innovation. And we believe that this will take the field of forward much faster way. And if you are in the precision medicine field, you'll be hearing from us in the near future. So future perspectives, precision medicine, has received very good press, and we have got the popular support and representation. I personally believe in a model of promising less and delivering more. But with this kind of press, we have a lot of responsibility on our shoulders that we have to deliver. So also in advancements in artificial intelligence field recently have presented a new promise to take the precision medicine to the next level. So deep learning algorithms now can diagnose diabetic retinopathy, skin cancer lesions at par with the physician. That means almost as well as physicians. And AI also interprets radiology reports with 91% accuracy. So this all so now the complex tasks of data analytics can be pulled into, can be done by artificial intelligence algorithms that will help, help with a lot of data analytics challenges that we are facing in the precision medicine field. And also, last decade belonged to medical products, equipments, hardwares, consumables. Current decade we are living in, it belongs to medical platforms. So we have, wearables, we have big data, we are generating a lot of data, and also health analytics. We're doing a lot of health analytics. But next decade will belong to medical solutions. So robotics, AI, augmented reality. What we can do with the data that we are generating in this decade. So that will be the challenge of the next decade. And also, if you see the figure, it presents an artificial intelligence market for healthcare applications worldwide. So in 2014, it was $663.8 million. But in 2021, it's projected to be $6.662 billion. So next decade belongs to finding medical solutions, and AI and robotics will define new health. You all may know about the government's All of Us initiatives. They're trying to create a research cohort of approximately 1 million American volunteers who will share genetic data, biological samples, and diet, lifestyle information, all linked to their electronic health record system. This will pioneer a new model for doing science, emphasizing engaged participation, responsible data sharing, and privacy protection, and all the folks who are interested in big data analytics and AI. For them, this will be a gold mine for innovation, and also trying to solve a lot of data analytics problems that the government may face. So future of precision medicine, according to me, is bright. We have moved, I think, from past symptom-based medicine to present, which is a pattern-based medicine, and to future will be algorithm-based medicine. In future, I think the drugs and algorithms will be launched together. That's what I sometimes to manage a disease. And so this is a little schematics that I have put where you can have patients once presented in the clinic, personal integrated omics will be performed, which plus the imaging data, which will be analyzed to create a personal molecular classifier, which will be which can be sent to a physician, and this is where digital health comes into the play, and the physician can see that and then deliver the promise of precision medicine, which is right drugs to the right patients and the right time. So whenever I talk about precision medicine, I always talk about safety and security. So Genetic Information and Non-Discrimination Act, which provides a strong protection to Americans against discrimination based on their genetic information when it comes to health insurance and employment. And again, these protections apply to residents of all 50 states and territories. However, GINA is not a comprehensive genetic privacy law, and it does not address all possible forms of genetic discrimination. So we need to do some innovation around that. However, GINA is a strong and essential first step towards precision medicine. So with that being said, I leave you with this final painting. It's called "The Doctor" by Luke Fields, and it presents concerned parents looking at a child who is sick and a doctor contemplating by looking at the patient. And what I believe that with the advancements in the field of precision medicine will provide doctors more tools and more tools to make their informed decisions to deliver the promise of precision medicine. Thank you very much.
Thank you, Dr. Mishra, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. All right, let's get started. What areas can AI be utilized in advancing precision medicine?
Yeah, AI is a very interesting field, and I'm very much excited to utilize AI in an early stage of drug discovery, where a lot of innovation really happens, to utilize AI to accelerate that the early stages of drug discovery. AI can be also utilized in running and monitoring clinical trials, and of course AI can when it comes to digital health and the data that we are sheer data that we are generating, I think to make sense of that data, I think AI can help our understanding and make our lives easier by analyzing all that data for us. So I think AI can help, I think, almost every aspect of data analytics that we will be using in precision medicine.
Okay. How can digital health tie into precision medicine?
That's an interesting question. So, digital health, well, now we call it digital health, right? But eventually it will be all health. So so we will have patients coming to, they can make appointments online. They can they can pay all to their physicians online on using their phones, and their symptoms can be monitored using the phone, so the digital health will become a really integrated part of the medicine in future. That's what I believe, and precision medicine will, and medicine overall will utilize advancements in digital health. And if you remember, the definition of precision medicine relies on lifestyle, lifestyle changes or patient's lifestyle. So to monitor that, I think that digital health, would be one of the very handy, to generate the data that's can be presented to a physician and a decision can be made.
Do you think AI would replace physicians?
Well, that's yes, some folks have different conflicting views, but what I believe I'm a very big advocate of not artificial intelligence, but intelligent augmentation. So I think the AI is a misnomer, but intelligence augmentation. So that so algorithms that we are designing, they're augmenting intelligence of physicians. So what we will do is, with the smart, like just an example, with the smartphones we became more smarter. We can make smarter decisions using some of the apps on our phones, right? So similarly, as with all these algorithms, AI will empower physicians, and some of the mundane tasks that are done by physicians, and we have that could be taken care of by AI, so that physician-friendly report can be submitted, where the physicians can take a look and make an informed decision. Sort of I mentioned the digital classifier. So so I think AI will become integral part of the medical system, but I don't think it will replace physicians. However, it will replace certain tasks that we do today. So I think I like the Stanford's quote, Stanford ran an article where they said that AI will, radiologists who will not adapt to AI will be replaced by radiologists who will adapt to AI. So similarly, physicians who will adapt to AI will replace physicians who would not. So I think that's in future we will see more physicians, learning, teaching themselves all the modern technology to help patients, to help patients and promise, and bring the promises of precision medicine to the clinic.
All right, we have time for one more question. How do we bring together all the stakeholders in the precision medicine field?
Yeah, that's one of the big challenges that we are facing right now. So we are trying to. So that's why we started the American Association of Precision Medicine, and I think with that we aim to organize conferences. We organize training programs for physicians, and we are planning to also bring all the stakeholders together to just foster communication and foster dialogue, and try to present problems. What are the problems in the field, and how can they be solved? And I'm a big fan of communications. Communications is magic, actually. When you put people in the room, solutions come out. So the communications will bring solutions. So I think we have to actively, as a community, invest in trying to bring the minds together and sort out solutions to these these problems that are caused by lack of communication. So I think if we build more platforms like that, this will be very beneficial as the community evolves further.
Excellent. Well, I would like to once again thank Dr. Mishra for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.